welcome to the Society of Chemical Industry. My name is Martin Elliott, and I'm a member of the group that arranges the SCI talks. This afternoon, our speaker, can we move on to the next slide, please? There will be a question and answer session at the end, and so please submit your questions at any time in the written form. Right. Okay, let's say my name is Martin Elliott, and I'm chairing this afternoon's session. Our speaker today, please move on to the next slide, is the founder and chief executive of Scottish Brain Sciences, Professor Craig Ritchie, who was until earlier this year Professor of Psychiatry of Aging at the University of Edinburgh and is now at the University of St Andrews. But he's a man who wears a lot of different hats, including director of the Centre of Dementia Prevention and a coordinator of the European Prevention of Alzheimer's Dementia Project. So with that, he's published over 400 academic papers, conference abstracts and book chapters, and secured over 100 million euros of grant funding in his career to date. And I'm sure that that won't stop there, will it, Craig?
I hope not.
Anyway, so with that, I shall hand the floor over to Craig, who's going to tell us about the brain health movement in the UK and as a life science priority. Thank you.
Thank you very much. Well, thanks very much, Martin, and thanks not only for the introduction, but also the invitation and the opportunity to share some thoughts. And I understand, of course, this is the third in a series of lectures that have been presented about dementia from James and Susan. Now, there's probably going to be, understandably, a little bit of overlap in what we present or talk about, but my primary focus is to really talk about this notion, this new concept we have of brain health as part and parcel of that kind of neurodegenerative process from early life right through to what we recognize as the dementia syndromes in later life. And also to dovetail this with, in some ways, my own personal journey as a clinical academic, as a doctor, and then as a CEO of a company, about why it is that we've decided to establish Scottish Brain Sciences to really try and move things forward a little bit more quickly and implement some of the research findings for patient benefit in a more speedy way than has historically been the case. This first slide just in a sense describes what Scottish Brain Sciences is, and then the next slide I'll overview the presentation I'm going to give. But I think this is an important holding point, if you like, that what we describe ourselves is one of the world's first translational medicine organizations. Now, translational medicine is quite a familiar word within sort of the medical community. But outside of that, it maybe sounds a little bit clunky. But in a sense, translational medicine is what we often refer to as bench to bedside. So that is, how do you make discoveries in a laboratory, and how do you translate them through the sort of that process of, discovery in a laboratory right through to being able to use in clinical practice. And when I set up Scottish Brain Sciences, the main purpose, the main objective of SBS was to accelerate that pipeline, that pathway from discovery to delivery. Now, there's many examples in medicine of innovations that have taken place that have taken quite some time. And in fact, on average, from a discovery being made, whether it's a diagnostic test or a new therapeutic, the average length of time from that discovery to being used in a patient is about 20 years. The exception to that was the COVID vaccine, which was obviously, months rather than years. But I think there's a long way to go. And I think as I'll explain, within the dementia space, I think that 20 years is actually 30, 40 years, and it's actually getting longer rather than shorter. And that was, I think, an observation, indeed a frustration, that led me to set up the company. So these are kind of just a few little dropping off points in the course of this presentation. What is Alzheimer's disease? And I know you've had presentations from both Susan and from James, But I think it's always useful to just sort of reframe some of or represent, if you like, some of the more basic concepts about these neurodegenerative diseases. And the next question is, what is dementia? Because I think in my next few slides, I'll explain the distinction between, AD, Alzheimer's disease, and AD, Alzheimer's dementia. And in doing so, I'll point towards, I think, the very near future about how we diagnose Alzheimer's disease, and rather than necessarily identifying someone with a dementia syndrome. Now, I'm not going to go into depth about the new treatments and new therapies that have been approved by the Medicines Health Related Agency, the MHRA. You'll have read in the press that these two new drugs, donanemab and lecanemab, have had MHRA approval, but here we are with NICE in England and the SMC in Scotland, still kind of deliberating about whether or not they can be used, particularly in the National Health Service. So I pose the question, should the NHS have been better prepared? And then finally, what is SBS at a more specific level, Scottish Brain Sciences, doing to ease access? So this presentation is kind of in 3 or 4 parts. The first is the empirical basis for this notion of disease before dementia, which comes from my academic world. Second part is about what we've done in Scotland to try and implement research. And then the final parts are about setting up the company and maybe some of my own personal reflections and challenges in that to try and accelerate the diagnostics and therapeutics. And maybe this will lead to a nice debate about whether or not this approach that we're taking with SBS could be a canary in the coal mine for other disciplines, other parts of medicine to emulate. So I'll go back one slide. Just to set the very, very, very basic foundation on this is that and I wouldn't be surprised if Susan and James had showed you similar slides, but on the left is the first patient with so-called Alzheimer's disease, and that's Alois Alzheimer on the right. She was very young when she was diagnosed. She was in her sort of late 40s when she started having symptoms, and she was in her early 50s when she died. And her husband gave consent, after she passed away, to do an autopsy. And then in that, Alzheimer discovered lesions which were not typically seen in somebody of her age, and those were the amyloid plaque, which is on the left, and these are his sketches from his lab book, and on the right was a neurofibrillary tangle. And these two lesions are pathognomic of Alzheimer's disease, and they go along with shrinkage of the brain itself and changes in the vasculature. But from 1907, when this was first described, until quite recently, the biggest challenge we faced in medicine was, unlike other branches of medicine, we couldn't make a tissue diagnosis because we can't really do brain biopsies. So we were having to rely almost exclusively on the phenomenological approach to diagnosis. And phenomenology means simply assessing the phenomena that emerge, if you like, the symptoms. Now, I'm a psychiatrist, and it's probably the reason why that up until 20 years ago, most people who were diagnosed with dementia were being diagnosed by psychiatrists in mental health institutions and clinics. It's because people were presenting with these diseases, with memory impairment, behavioral challenges, neuropsychiatric symptoms like depression, hallucinations, anxiety. And those were the kind of, in psychiatry, we're very, very used to. In fact, it's almost always the case that we make diagnosis and symptoms. There's no blood test for depression. There's no brain scan for schizophrenia. So this fell within our sort of our field of expertise. I think what it did do, though, however, was create a misconception of what Alzheimer's disease was. And what this figure demonstrates is that we started in the 20th century by describing dementia, not just in the clinic, but also in the diagnostic manuals, as a memory disorder, as a cognitive disorder. So we were actually describing these brain diseases on the basis of the symptoms that emerged, which we now know to be actually very late in the course of the disease. So where we've also had to therefore do our research is been in the people who present to our services, and those people present late. So this notion that these neurodegenerative diseases are diseases of late life, characterized by memory symptoms, is actually incorrect. These diseases, the Alzheimer's disease, for instance, is the main example because it's the most common pathology that causes dementia. Originally, 10 years ago, when we started doing some cohort studies, we hypothesized that these diseases actually started in midlife, and they expressed themselves with these typical symptoms that we call dementia in late life. But there's a 20, 30-year period when these diseases were developing in the brain before traditional symptoms emerged. So I'm going to show you some data from one or two of the studies that I've been fortunate enough to lead in the UK and the other across Europe, which provided, if you like, the empirical basis for why we are so confident now that these diseases start many decades before dementia develops. So the important thing with that knowledge wasn't just to publish it and to get grants and further accolade about how clever we were at discovering these things. The most important thing was how can we implement those findings into clinical practice, and I'll talk a little bit more about how we did that in Scotland. But the argument being, like with any other branch of medicine or any other disease, the earlier you can pick up the disease, the more likely it is that you're going to be able to affect its course, either through risk factor modification or indeed therapeutic intervention. So in doing so, if we can detect these diseases early, then ipso facto, we prevent dementia. So I've often been quoted as saying that I think dementia itself will be a very, very rare condition within the next 5 to 10 years. Much as my wife is an infectious diseases doctor at UCL, and she specializes in HIV medicine. Now, very few people, I asked her this the other day, 0.02% of people with HIV in the UK will develop AIDS. Now, HIV is still, reasonably prevalent, but no one who's diagnosed with HIV in the UK would be expected to develop AIDS. So this should be the same sort of approach that we take to neurodegenerative diseases. If we detect them early enough and we manage them effectively, it would be a failure on the part of medicine if that person goes on to develop a dementia syndrome. So one of the projects that I was fortunate enough to lead that kind of centered itself around these concepts was the European Prevention of Alzheimer's Dementia Programme, which is funded by the Innovative Medicines Initiative, IMI, which is an explicitly public-private partnership run by the European Union, and actually brought many of the top-notch academic universities in Europe alongside some very large pharmaceutical companies who have got a particular pipeline or interest in Alzheimer's disease, with other organizations like SMEs and charities like Alzheimer Europe, etc. And the purpose of EPAD was to develop what we call a trial platform, a clinical trial platform, where we could test much more quickly, much more effectively, much more accurately, series of compounds that would affect the disease course in a population who did not have a dementia syndrome, hence the prevention of Alzheimer's dementia, the European Prevention of Alzheimer's Dementia. Now, the whole project was actually premised on the ability to develop what we call the longitudinal cohort study. And that cohort study had 2,000 people in it, some of whom are still being followed up. And in that cohort, we were also able to capture really important information, data on many biological processes taking place in the brain and relating them to genetic factors, lifestyle factors, etc. And unfortunately, when we were about to initiate the trial platform, it was 2020 and COVID hit. So we lost 2 years of momentum, and the grant concluded, and sustaining this became almost impossible after COVID, in 2022. And this is another catalyst for me, because I was the academic leader of this program, to set up a company, because I felt the principles that underpinned the EPAD program were robust, but it was unlikely that we were ever going to be able to do anything of this scale at a kind of a publicly funded level. So rather than miss the opportunity, I've spent some time in learning about what we could have done better, and embedded that within some of the objectives and the business structure of Scottish Brain Sciences. I think one of the other things about this program is that the data that's been collected on this cohort study is freely available on a platform called ADDI, which we helped design, Alzheimer's Disease Data Initiative. And this is actually funded almost exclusively, I think, by Gates Ventures. So what this allowed us to do is set up a data platform, which is incredibly easy to access, and very quick to access. So I don't expect everyone on this meeting to know some of the challenges with data access in the medical world, but if you wanted to access, for instance, biobank data, UK biobank data, by the time you put your application into actually having the data in a place you can analyze, can take several months, if not even a year. Whereas with the systems that we set up in EPAD, it takes 3 hours. Because there's no application process. It's assumed that if you have an academic university email address, then you're a valid researcher, and therefore you can get access to these anonymized data. And it's also free, and that's why it's been really important to us. And this is actually an old slide. This is probably about 6-9 months old. That a lot of the data access has taken place from low and middle income countries. So Africa, South America, Southeast Asia, etc. And I think the democratization of this data is actually really, really important, so that other academic groups can benefit from it. The other project I'll talk about is the Prevent Dementia program, which was originally funded in 2013 by the Alzheimer's Society. So this preceded EPAD by a couple of years. This study wasn't affected by COVID to the same extent. It's still ongoing. In fact, yesterday I was in Dublin. We have an annual General Assembly of all the academics and researchers working on the program. There's about 50, 60 of us in the room talking about some of the data, some of the science that's coming out, but also planning the next 10 years. The original cohort has 700 people in it, aged between 40 and 59. So there's this midlife group that I was referring to earlier. And they've been followed up now. We're now on the third wave of follow-up. And we enriched the population with connections that I was very fortunate to make with individuals in Scottish rugby. So we were able to get some grant funding to look at elite rugby players, professional rugby players. It's public knowledge that that Shane Williams from Wales and Ben Kay from England are part of this cohort. They've tweeted about it. I think we've got 4 World Cup winners in this, and we're delighted to say, actually, of the 60-odd elite players who are in the cohort, they're almost equally distributed between men and women. So we're able to look at some of those differences, gender differences in contact sport. And on the basis of that, we got a grant from FIFA and the FA to look at elite football players, and we're still currently recruiting them. So this is actually, in some ways, a high-risk population within the general population to be able to explore, risk factors and their association with changes early in the brain, and how those over time, with the follow-up, emerge into symptoms and perhaps some clinical problems. So just thought I'd give you the sort of the headlines of those 2 programs before I jump into some of the data, because it's from those projects that these data come from. I think one of the things that is worth highlighting is, as I mentioned earlier, when we talk about a dementia syndrome, we talk about Alzheimer's dementia, we always tend to think of older people with clinical symptoms like memory impairment, then may go on to other cognitive challenges like planning, judgment, speech, language, etc. We believe from cohort studies, but also from post-mortem and pathology studies, that the area of the brain that seems to be very susceptible to Alzheimer's pathology early on in the course is a region of the brain called the hippocampus. So it stands to reason that in the early phase in early stages of life, could we test specifically the hippocampus to see if it's beginning to fail? Many years, decades even before those kind of gross signs of brain failure start to emerge. So there's two ways that one can do that. Number one is through brain imaging, and we can look at hippocampal subfields, and these are parts of that hippocampal structure that was in this slide, broken up into smaller areas. And each of those smaller areas subserves a particular function from a cognitive or a behavioral perspective. And some of them, areas called CA2 and CA3, are very much involved in what's best described as visuospatial memory. There's areas, this is Nobel Prize winning work from UCL, about how these cells within these regions of the hippocampus are involved in orientating ourselves to the environment around about us, easily described as visuospatial function. So the question then arose, well, is there a way that we can test the function of these hippocampal place cells, and the other ones are called hippocampal grid cells, in a memory test, or sorry, I shouldn't say that, a cognitive test? And if I were to, do a cognitive test typical of what we would do in a memory clinic in older people, everybody on this call, everybody who's aged between 40 and 59, would get or approach getting 100% in the test. So you don't learn anything, because these tests have such a, what we call, high ceiling effect. So what we needed to do was actually develop tests that were specific to this region of the brain, and also were hard. They were tough tests. And one of them is what we call the Four Mountains test. Now, what I'm going to show you, and you can all do this, is I'm going to show you a picture of four mountains. And these four mountains obviously have a particular orientation to each other. Then once you've actually, looked at these for a while and you've, stored them in your memory, I'm going to show you another slide that has four choices on it, only one of which is the same four mountains from a different perspective. And we call this allocentric visuospatial memory. You're kind of outside the scene looking in, rather than being inside the scene, like being inside a supermarket, for instance, sort of looking out, and that's called egocentric visuospatial memory. So if I just show you this slide, you'll see these four mountains, 1, 2, 3, 4, this little one here, here, here. And what I'm going to do in a minute is actually show you the same four mountains, but from a different perspective, and actually a different time of day as well. But there'll be three other four mountains arrayed against each other, only one of which is the correct one. So when I do this in a live audience, I can get people to show their hands to vote, whether it's A, B, C, or D. But you can see in this, it's much harder than a test you would get in a normal memory clinic about what's the date, or what's the name of this building. And to put out your misery, the answer is A. So that Four Mountains test was something that we've done in the Prevent project. And what we showed, and this is a friend and colleague, no relative, Craig Ritchie. She's from Tasmania originally, now works in Montpellier, very famous neuropsychologist. And what she did was she looked at the Prevent dementia population from the first site where we did this project, which was in Imperial College, and actually the West Middlesex was our centre, for those who know the area. And these people were asked to do the Four Mountains test. They were also scaled or rated on something called the CADE score, which is a measure of dementia risk in the future. It's been well validated by another friend of mine, Mia Kivipelto, who's based in the Karolinska. And people with a higher CADE score are more at risk of developing dementia in the future than those with a lower CAGE score. So it's made up of weight, age, sex, education, etc. And lo and behold, that even at the age of 52, on average, mean age of 52, people were already scoring lower, quite significantly lower, on the Four Mountains test if you're at a higher risk of dementia in 10, 20 years' time than those who had a lower risk. So here already was evidence that the risk for dementia was manifesting itself in a subtle, but probably reasonably specific effect on hippocampal function. So complementary to this, Mariolina Donavi, who works in Cambridge, in our imaging group, is based at Cambridge University under Professor John O'Brien, and I know James Rowe, who you heard from in September, is also from Cambridge, same kind of research group there. What she did was actually, rather than look at function, she looked at structure. So she now looked at a genetic risk for Alzheimer's dementia, something called ApoE, apolipoprotein E, which if you're up to date with celebrity news, you may know that Chris Hemsworth, who plays Thor in these movies, recently got tested, I think on 23andMe, and discovered he was an ApoE4 homozygote. So he had an ApoE4 allele from his mom and his dad. And that puts you 17 times at risk of developing dementia in the future. So there's the same gene here that Mariolina was looking at. And what she discovered was that certain areas of the brain in this cohort from West London were smaller, again, mean age of 52, in people who had carried this ApoE4 gene. So there's already an interesting finding here. And these were areas, by the way, that were, like I say, CA1 and CA2 and 3 here. These were areas of the brain which are known to be early impacted upon by Alzheimer's pathology, amyloid and tau. So again, good empirical evidence that something's changing many years before traditional or characteristic symptoms of dementia develop. Now, there's over 100 papers, maybe 150 papers published from the PREVENT and the EPAD program. They're all kind of pointing in the same direction. One of the big findings, and this is a reasonably complicated slide, so I'm going to walk, or study, so I'll walk through it reasonably slowly. But one of the assumptions, and I think it's proving to be true, is that the aggregation of amyloid, this protein that sits outside the nerve cells, which we're all developing, we're all aggregating it. It's a normal process in life, but for some reason in people with Alzheimer's who go on to develop Alzheimer's disease, you get more of it developing, and you also get a more toxic species developing, which is synaptotoxic and neurotoxic. This protein amyloid is thought to precede the aggregation and spread of tau. And what this little cartoon here is demonstrating is that if you go to the bottom line, then the symbol, orange for elevated, blue for decreased. And this here, ApoE4, means that tau is elevated and tau is decreased in the orange and the blue. Now, we think that tau is what really drives symptoms. The amyloid itself triggers the tau. It's the spread of tau which really damages the nerve cells and the synapse. So if you're amyloid positive and you're female or you're male, doesn't matter, and you're ApoE4 carrier, male or female, you get an elevation in tau. That's normal. That's what we always thought was going to be the case. The interesting thing is when Tyler, who was one of my PhD students, looked at the amyloid negative people, so these are people with lower levels of amyloid in their brain in the EPAD project, a very interesting thing emerged. If you're amyloid negative in male, then you have no difference really in whether you're ApoE4 positive or negative in your tau status. Amyloid negative, tau negative, nothing to see here, move on. If you're amyloid negative and you're female, however, and you carry the ApoE4 gene, then you actually see an elevation in tau. Now, this is actually incredibly important because what it implies, and this is, like I said, we've got other evidence to support this from other studies, is that, hold the front page, men and women are different. And what we're seeing is we're seeing an emergence of this difference in earlier stages of disease, When you look at males and females at the end of the disease process in dementia, you don't see these differences. Everything comes together. But what this would imply is that to elevate tau, if you're female, you don't need the same levels of amyloid. So you're maybe more sensitive to whatever amyloid is there, and you generate tau on that basis. Now, this is really important for therapies because we tend to treat people now with these new disease-modifying therapies on whether they're amyloid positive or negative. Now, for a woman, the threshold for amyloid positivity may be a lot lower. So if you're using the kind of the general threshold, then a lot of women are missing out on therapy because they haven't got the same level of amyloid on average, which would trigger treatment eligibility. There's also emerging evidence that there's quite significant ethnic differences in this interaction between amyloid and tau. And African Americans, for instance, have this similar picture where they seem to be able to develop dementia and tau pathology with much lower levels of amyloid. So this is actually, critically important. So when I first presented this data in a meeting, I was in Australia, I was in Sydney, and I thought this was a good river system to illustrate the point. So as I said earlier, if you're only looking at the late stage of the disease, you're imagining yourself up at Sydney Harbour Bridge. You're in the estuary. You don't know where that water has come from. Has it come from the Middle Harbour River, or has it come from the Parramatta River? Has it come, from a woman? Has it come from a man? It's all there, all joined up together. So what we needed to do, and excuse the pun, we had to go upstream. Going upstream in a disease process means going earlier in life. Go back in time to an earlier population. And what this was demonstrating was that men and women were two different rivers. And then we had to start thinking about whether or not, when we analyze data that we collect from people in midlife, should we use gender as we typically do statistically as a confounder? Should we use it as an interactive term? Or should we do what we're doing now, which is actually stratifying the sample into two populations? So these were important findings that we would never have made had we continued to only do research in older people and try and impute our findings back to a younger population. Now, the next thing that was, I think, quite interesting, if you remember, Mariolina demonstrated that this ApoE4 gene was important. It seemed to have an impact on hippocampal volumes in midlife, which was maybe manifesting as changes in that Four Mountains test of visual spatial functions. So we were in a position thinking ApoE4 is bad, it's not good for your brain health. But actually what we've come to understand through further analysis of our data, is that ApoE4 actually seems to be beneficial for brain health up until the age of about 52. So generally speaking, ApoE4 seems to have a positive effect on brain health. And one of the examples here is, apologies for all the shorthand, but these are all the different cerebrocortical arteries: anterior cortical artery, middle cortical artery, cerebral, middle cerebral and posterior cerebral artery. And this is the amount of blood flow. You can measure blood flow through a technique called arterial spin labeling, ASL. So this is you can see the blood flow is greater in each of these big arteries feeding the brain if you're ApoE4 positive, right across the board. So it seemed, and there was other evidence about connectivity and other things that were demonstrating that ApoE4 seems to have some beneficial effect, and then is lost for some reason as one gets older. Now, I think this is probably the last academic paper I'm going to share with you. But this paper, which came out in January of last year, in my mind was one of the most exciting papers I've ever been involved with. Number one, it was using the EPAD data, which we generated, but not by somebody who's part of the consortium. So Rasha Saleh, who's at the University of East Anglia, got the data access and did her analysis. And one of the other reasons, not only is it a really powerful finding, but it really did look at this interaction between women's health and brain's health. And what she looked at in EPAD was the 1,074 women who were in EPAD, and she split them by their ApoE4 status. And then she looked at what impact HRT had. And these people were mean age of 62, so they were all postmenopausal. So she had a look and said, well, what does being on HRT do to your brain health relative to your E4 status. Now, if you recall what I just said, this genetic risk, the Chris Hemsworth gene, that increases your risk of dementia 17-fold if you're a homozygote and 3 or 4-fold if you're a heterozygote, is bad for brain health in the dementia population, but seems to be good for brain health in the younger population. So what she demonstrated was that people, women, who carried the ApoE4 gene, remember, possibly good for brain health up until the age of about 50, who then went on to HRT, had the best brain health. So it seemed to be, if you took the headline on this, that HRT maintains the benefit of ApoE4 beyond the menopause. So I know this is a very busy slide, but this was true both of a cognitive measure called the R bands, which is a kind of a global measure of cognitive function. So here you have people, yes, taking HRT, no taking HRT, green and blue respectively, non-E4 and E4. And you can see the best condition, the biggest difference there is in the E4s who are taking HRT. And this is more obvious with a particular memory test called the delayed memory index. And the same is true statistically of some brain volumes that are important, the right entorhinal and left entorhinal volume. There was also some evidence that the sooner you took HRT after menopause and the duration of taking it were also beneficial. So to my mind, this was quite compelling evidence that we kind of almost like reinforce this belief ApoE4 may be good for you up until the 5th decade. And then women who take HRT, this benefit is maintained. So really interesting data. So, and it completely throws in its head things that we would have thought we knew in older people if we only studied dementia. So, or the dementia syndrome. So there are both cognitive and structural changes in the brain that represent the AD signature, and there are important sex differences. And ApoE4 may confer resilience and better brain function up until the 5th decade. I'm just aware of the time, so I'm going to flick through because I want to leave, I think, plenty of time for questions. So maybe just another 5-10 minutes of presentation. So we wrote this paper in 2015. Craig Ritchie and myself asked the rhetorical question, is late onset Alzheimer's disease really disease of midlife? And I think now nobody on this planet would disagree with this. I think that the evidence now is so compelling from our studies and other studies related to it that these are diseases that have a very long prodromic period before dementia syndromes arise. So in 2019, I met with the First Minister here in Scotland. I moved back to Scotland in 2014. And we set up something called Brain Health Scotland. And that was very much about arguing the research was mature enough that we should actually be putting it into practice as quickly as possible. And that led to a government-backed initiative called Brain Health Scotland. Henry Simmons is the CEO of Alzheimer's Scotland, and my friend Anna Borthwick was the executive lead of Brain Health Scotland with me as the inaugural director. One part of the Brain Health Scotland project, if you like, was setting up what we call brain health clinics. And these are quite different to memory clinics. Memory clinics are there to make a diagnosis of dementia and provide post-diagnostic support. The 3 pillars within a brain health clinic are early detection, risk profiling, and then putting in place a personalized prevention plan. It's a very busy slide. Just to walk through it, these are community accessible services. Only one exists in the NHS, which is in Aberdeen. I do a clinic like this for ex-professional rugby players at Murrayfield, the same model, but outside the NHS. And it's all about, like I say, risk profiling, putting in place tests for early detection, and then putting in place a personalized prevention plan. And in the NHS, there's a kind of various triage steps as you go deeper and deeper into the assessments. The rugby clinic at Murrayfield, it's a one-stop shop. I do everything on everybody, simply because they're a very high-risk population. So I think that they would always make it through all these triage points if we put them in place. Now, just for the last 5 minutes, I just want to talk a little bit about life sciences, because I read this report called the Campbell Report, which came out in 21. So this was about a year before I set up Scottish Brain Sciences. And it really articulated for me a lot of really important points that it really wasn't possible to solve or to deliver on a lot of the promise that new sciences was pointing towards if we relied exclusively on the health sector or exclusively on the university system or exclusively on the life sciences sector. And it was only through this notion of the triple helix, where the academic institutions, the healthcare systems, and the private sector all come together to not only drive better health outcomes, but also concurrently increase economic growth and drive inward investment. It seemed to be a no-brainer that if you were able to implement and innovate on health outcomes, you should be able to also derive economic growth and inward investment. And I was, thought, well, maybe the topic that we're on today about brain health would be a good exemplar of how we can actually push this forward. So I did speak to, partners in the Scottish government before I set up the company about how this dovetailed, if you like, with the ambition of the Campbell Report. So this little arc here, this is almost my narrative arc, was that we started off with Prevent and EPAD, providing the scientific foundation. We spent some time with Brain Health Scotland, trying to set up the implementation of the science. But in the NHS and in the charity sector, it proved very, very slow, glacial even. So on the foundation of science, the implementation of Brain Health Scotland, what I say is the Scottish Brain Science is about acceleration. It's not doing anything different. It's not having a different objective. It's trying to do it much more effectively. So I think I've covered a lot of these points, but what I haven't really talked about in detail is what does SBS do to ease access to these treatments and trials. So I turn now to corporate speak, where, away from the academic and the clinical to what is investable in, what is something that can be, from a business to business perspective, can be seen as worth partnering on. So let's imagine a war on dementia, which I've been trying to win for 25 years almost. Break it down into missions. So we want to develop the tests and the treatments to identify and then cure. And I truly believe we can cure these diseases. But then the next mission is, that's all good and well to have these tests. You have to implement them into the brain health services as we've designed historically. And then you have to think about, from a commercial perspective, how can you maintain this machine of implementation? And that means looking at the data set and the biobank that you collect in doing this and using that for commercial purposes. And the final mission is, because we're incubating this in what is a very nice little country, but it's a very little country, is how can you take the innovations and the ideas out of Scotland to the rest of the world? And of course, as a company, we're not bounded by geography. So at the core of this is, if you remember, I've talked now about the Prevent cohort and the EPAD cohort. Well, here's my third one, the Iona cohort. So this is sponsored by Scottish Brain Sciences, and it's a huge cohort already of individuals where we very deeply characterize their brain health through brain scanning, through blood testing, through spinal fluid, through cognitive evaluation, and all the risk factors. And this allows people to come into clinical trials that they may be suitable for. It allows us to develop diagnostics. It allows us to act as a platform for drug discovery, and it allows, by feeding back all their test results, to us providing healthcare through a research project. And the clinical, the academic benefits, I should say, of this database are for better disease modeling to complement some of the stuff I've shown you about Prevent and EPAD. Ultimately, to be able to put in place, probably through machine learning and AI approaches, a risk prediction tool, so that we can actually say well ahead of time what the risk of developing X, Y, or Z is. And then also as a potential for actually working with other partners, be they academic or commercial, to drive future research. Now, this is a rather cheesy photograph, and I apologize for it. But one of the things that we did differently also in Scottish Brain Sciences was say, we're not going to have to wait for the NHS to gatekeep referrals of people to us. So what we did was we went direct to the public. And the reason I show this is, I mean, I don't expect anybody here, even if they've lived in Scotland, to know, but about 18 months ago, there's quite a big profile piece about me and the company in the Times. And it led to the headline, which I didn't expect, about dementia expert quits the failing NHS. It led to actually a question in the House in Holyrood about how the SNP government let such an idiot leave the NHS. But anyway, what it did do was drive huge public interest in what we were doing in SBS, and it drove really recruitment. So one of the things I think with our public engagement work, and there's me, and I mentioned earlier that, you know, with the Deputy First Minister. So what we've done is we've actually really engaged the public as our main kind of partner in a lot of what we do. And these are just some public engagement events. And I mentioned earlier that when I was chatting to Martin earlier, there's actually a BBC documentary with Sally Magnuson that went out about four weeks ago, which is now an iPlayer, about the company and the approach and all the things we've been talking about. So in some ways, what this points to is this triple helix of the health sector and the universities and the life sciences sector, actually adding an additional, probably the most important strand, which is the public, and that's the quadruple helix. And it's working. I mean, we've been recruiting into this cohort study at, I think, 3 times faster than we did in EPAD, with 64 million euros for EPAD. In a commercial sense, going direct to public, we've achieved much more with a hell of a lot less in terms of funding. And we're already beginning to recruit rapidly into clinical trials, which is obviously driving, in some ways, inward investment from the pharmaceutical companies to set up trials in the UK, in Scotland in particular. And that gets us out of a hole, because the NHS has been, dare I say it, challenged to be able to get people into trials other than possibly cancer. So, and we're doing well in terms of the perception of us from the public. So these are just sort of staff surveys of our participants, to make sure that they're getting a good valued service from us. And very simple principles. You said, we did. So making sure that when they come into studies, they want to stay, because they have good experience. Because I've often said that it's much easier to recruit people than it is to retain. So we have studies, the Iona study will have an annual visit going on forever. The last thing we don't want to do is recruit 1,000 people and only 100 to turn up for their follow-up visit. The reason I show you this picture is this is the second floor here is where our office is going to be in Aberdeen, which people say, this is beautiful. This is not the NHS, is it? And I think these are where we're going to do our infusion suites, and we're going to do our early testing, et cetera, So people will want to come and have a visit because of the quality of the hospitality that's on offer. So my last slide is that, given the scientific background, given the implementation in the NHS and the public sector, established on the basis of that knowledge and those learnings, the world's first translational medicine organization, taking on board a lot of the pain points that we'd been through over the previous 20 years, which are understandable, but that doesn't mean to say we had to accept them. We deliberately blurred the boundary, like I said, between clinical care and clinical practice. That should actually be clinical research and clinical practice, not clinical care and clinical practice. We leave nothing to chance. So we've got our own imaging facilities, our own laboratory, our own infusion suites, etc. We don't accept the unacceptable, which was things beginning to happen in the NHS, that people were kind of like down, lowering their horizon for what they thought was good service. And I'm very aware that Scotland's a tiny country, but it's a good place to incubate ideas and work very closely with our business partners, but make sure that we always prioritize what our key investors' needs are. And that might sound a little bit commercial, but I consider our biggest investors are actually our participants. And that's why they always come first in what we do. So I'm going to stop there, and I apologize, I did run over a little bit of time, and really look forward to questions or further discussion.
Well, Craig, not very often I'm accused of being speechless, but you've left me with so many questions that one of the things I know I'm going to be doing as soon as it's up on the YouTube is rewatching that again. It's been absolutely fascinating.
Thank you.
it started off by, if you like, you put me through a range of emotions there.
Ah, okay.
From the starting off point where you said that it's taking 20 years of development time,
Mm,
I'd see that going out to 30 to 40 years.
mm,
Now, when I was working in the pharma industry just after I'd left uni, at that time, and that this is, shall we say, almost half a century ago, they were saying then that it was 20 years of
yeah,
development.
that's changed. Yeah.
But then, thought of that doubling going out to 40 years, where we've got things like AI coming into the world.
Mhm.
It's rather depressing. But
Yeah.
you took me through as I say, the range of emotions. I've left the end of this, feeling, very, very positive about the way that this subject is moving forward. So
Mhm.
a couple of questions for you.
Yeah.
How have you found fundraising?
Um, in the commercial sector, much harder than it used to be in the public sector. I think one of the biggest challenges has been trying to describe the business model, because we don't fit very easily into any particular box. Investors, venture capital are, they understand clinical research organizations, they might understand a site management organization, they might understand a data sciences company. But when you try to do a little bit of all of that, then that's probably where you have to be much better at communicating. What is a translational medicine organization? How's that different and how's that investable? And I think the other thing, of course, is, well, what's your revenue stream? How are you going to make money out of this business? So that's something that we're always working on to improve that communication. And we're also 2 years old now. We do have evidence of it working, so we should be easier to get the Series A funding, which is kind of planned for next year.
Well, all good fortune with that. Now, Will Dean from the audience has asked, what do you feel needs to happen in the awareness raising aspect of brain health and dementia?
That's a great question. I mean, when we were at Brain Health Scotland, we did some Ipsos Murray surveys. One of the first things we did was we went out to about 1,500 Scottish people across I think 20 to 39, across socioeconomic and ethnic different groups. And one of the questions we asked was, do you have concerns about cancer? Do you have concerns about cardiovascular disease? Do you have concerns about brain health or dementia? And interestingly, about 75% of that population said they had concerns about all three of those. So it was an equal level of concern, in inverted commas, about those three major conditions. But then when we asked them about what actions can you take to maintain your brain health, the numbers were incredibly low. Only 3% of people thought that a head injury was associated with dementia. 3%. So what we recognized was there was a huge knowledge gap between concern about brain health and dementia and actual knowledge about what steps one could take. So that was almost from a public health perspective. I think the first step is closing that educational gap. So we put in place a lot of programs here in Scotland, including with school children. We've got a program called the STARS program, which goes out to 10-year-olds in school as part of the curriculum here in Scotland, about factors associated with improving or maintaining brain health. So I think there's something called the Lancet Commission report, which identified 14 modifiable risk factors for dementia. And if you just go through those at a public health level and an individual level in a clinic, then you're going to really move the dial, because they think that will probably reduce the incidence of dementia by 40% if all of those risk factors were brought to zero. So I know it's a very long answer, but I think what we need to do is just help the public understand that there are actions they can take that will be good for brain health. And we never talk about dementia prevention. We talk about brain health. What's good for your brain? Diet, nutrition, sleep, avoiding, or managing hypertension, high cholesterol, these types of things.
Thank you. How do you feel that the change of what's happened in Westminster in the last 6 months with the change is going to impact upon this environment?
Well, I think the Look, I think some of the statements, the public statements that have been made do seem somewhat familiar. I mean, when the Darcy report came out and was obviously highlighting the need for prevention and be health related rather than illness related. Well, I can remember Ara Darcy saying that 10, 15, 20 years ago. I think we've known for a long time that the health system, well, I'm not even going to call it the health system, the illness system is buckling under the strain because we don't do anything to maintain health. The National Health Service, it doesn't really exist. It's a National Illness Service. I think 2% of budget is spent on public health, 98% on acute care. So you can see where the balance of funding goes. And that's why we have the front door for healthcare for most people in this country is the emergency department, which is probably the worst place in the world to be at the front door of your health concern. So I think the Labour Party have obviously prioritised the National Health Service, but as we were chatting about before the seminar started, I actually think that the public deserve an open dialogue on whether or not we should be evolving the National Health Service or creating a new National Health Service for the 21st century. And that doesn't mean to say we start from where we are now. We might want to start from a very different place. And what I'm doing, in effect, with Scottish Brain Sciences is very, very visible to Scottish Government and the Health Minister and the Deputy First Minister, etc. Now, I know this is going out to the public, so I'm not going to make any wild statements, or on YouTube, but they're interested in this model of actually using the kind of the triple helix as a way not only to drive the economy, but also to provide healthcare. And I think that's the thesis behind the Campbell report. So I think unless the Labour Party embrace the values that come from the life sciences sector and really see them as an equal partner, then I think we're going to be stuck for a very long time.
Thank you for that. Well, I think that that brings us to the end of this afternoon's questions, unless anybody's got anything at the last knockings. But whilst we're waiting to see whether anybody comes in, can I bring your attention to the next trio of talks, which are on the question of sustainability. February, March and April of next year, looking at the carbon economy, the good and bad and the ugly of life cycle assessment, and then finishing up with the role of advocacy in accelerating the transition to more sustainable chemicals. And no, I think we've come to the conclusion of this afternoon. And so, Craig, I'd like to thank you very much indeed.
Thank you.
It's, as I say, it's taken me through an emotional roller coaster. It's left me with the desire to be rewatching this this talk as soon as it appears up on YouTube, and I would encourage others to have a look at the SCI YouTube channel. I'm sure you'll find a lot of things of interest to you in that time. So many thanks for your time, everybody, and I wish you all well. All the best. Bye bye.